All right, good afternoon, everyone. Welcome back from break. We're going to go ahead and get started with the second half of our program. So as we move into a discussion of current and future treatments for chemo-induced hearing loss, one of the things that always comes into the discussion is the fact that those new future products need to be developed and tested in clinical trials. And so to kind of serve as background for our discussion with all of you, we have Dr. Greg Aune, who's going to provide a presentation on challenges in clinical trial design in chemotherapy-induced hearing loss. Dr. Aune is a physician and researcher with training in pediatrics and hematology-oncology. He's also the assistant professor at the UT Health San Antonio-Greehey Children's Cancer Research Institute. So please join me in welcoming Dr. Aune.
Yeah, thanks. I think it's a pleasure to be here, and so far I've really enjoyed the discussion. And for those of you that have spoke up, I just truly applaud the bravery that I think it takes to come and tell your story, and it's going to, in the long run, make a big difference. So I get the opportunity to talk about how some of these late effects impact our way that we develop clinical trials. And so I wanted to start off by talking about one of my favorite patients. I think I went the wrong way. So this is a patient in San Antonio that's well known to our service. He had Hodgkin's disease in the early 1990s. His treatment then included splenectomy, a lot of radiation, 8 cycles of chemotherapy with 8 different drugs over about 9 months. And the treatment was also in that era didn't have a lot of the supportive care medicines that we have at our disposal now, and so that impacted, I think, some of the acute side effects that the patient experienced. So fortunately, this patient did survive. In the early era of the 1990s, I think Hodgkin's disease was even then was over 90% survivable if you looked at a 5-year survival rate. And then he went on and has lived from over 2 decades, and through that time has developed a lot of different health problems, which I think are probably not a surprise to anybody here, but I always tell people this is a fairly typical survivor story in that there's multiple treatment-induced effects in survivors. So this patient has experienced hypothyroidism, pulmonary fibrosis causing a couple of lengthy ICU admissions for pneumonia, infertility, fairly severe cardiac disease, including aortic stenosis, 3-vessel coronary artery disease, which required open heart surgery in 2008. The patient experienced a couple secondary skin cancers, a mild stroke, which left no permanent disability, and then also has developed diabetes. Most of these, if not all of them, are secondary to the treatments that were received for his childhood cancer therapy. And this is one of my favorite patients because it's actually me. That's my story as a survivor. And that is my family in Hawaii last June. So I mentioned that I suffer from infertility, but I have 2 sets of twins from semen that was banked before I was treated with chemotherapy. I think being here today is very special to me because, like many of you, I'm a survivor of cancer, and I'm a physician and I do research now. But I think first I was a patient, and I know kind of the struggles that we have to go through as survivors, and I think there's hope for the future. There's hope for all of you here, is what I would say. Cancer was the worst thing that ever happened to me, but it was also probably one of my biggest blessings because I think it just put me on a different track in life. I've had to overcome a lot of obstacles. They can be overcome, and you can do great things. So that's kind of my motivational message, is that I think whatever you face, you can overcome it. I think the point of today is really trying to think about how we can help those of us affected by late effects in general, overcome the obstacles, and then look to the future and take our experiences and use them to better outcomes for future patients. So I've had a lot of late effects. I know a lot about late effects. The one late effect I don't have is ototoxicity. My wife might argue with that. But I did have my hearing tested a year ago. She thought I wasn't hearing, and it was normal. So I don't know about ototoxicity. I don't personally experience that, but I know about late effects. And I've thought a lot about what kind of problems this poses for our clinical trial design, because I think it's really important as we go forward that we try to solve this problem. And so we're all well aware of the success of oncology, pediatric oncology. And I always just put a graph up that shows our gains in survival over time. This is from a review article published by Peter Adamson. The last bar on there is survival, I think, in 2004. And at that time, 5-year survival was approaching 80%. We're not over 80%, I would say. But what a 5-year survival says is whether a patient is alive or dead after receiving their diagnosis 5 years later. It doesn't say anything about their quality of life. And I think what we've seen here today is quality of life is becoming an increasingly important issue in pediatric oncology that we must consider. And so in the 21st century, childhood cancer, I think, is at a critical point. Our survival rates are greater than 75 to 80%. We still do have a large issue with patients not even surviving, with childhood cancer still remaining the #1 cause of death by disease in the U.S. But because of our success with the other 80%, our survivor population is booming. We're going to have over 500,000 long-term survivors of childhood cancer in the U.S. by 2020. Kevin Effinger, in a paper in 2006, I think, in one of the seminal studies that was done, showed that of this population that's growing, approximately 1/3 or greater can expect to have at least one life-threatening late effect as the result of their previous cancer treatments. So we're kind of in a new era. I wanted to talk a little bit about how we do research, preclinical research, that leads to our clinical trials in the era of that we face right now. So historically, on the top, this was supposed to all go in sequence. I think my keys got messed up, so I'm just going to use my pointer here if I can figure it out. Historically, for drug development, it's focused on creating drugs that target tumors, and we use a combination of testing potential drugs on cancer cell lines in culture, then moving into mouse models where we test candidates in mice that have tumors, and then we move into phase 1, 2, and 3 clinical trials. With late effects, the way we've actually done the research is we give patients standard of care treatment, and then we wait decades. And then we organize cohorts of patients that are survivors, and we look for health problems. So things like secondary cancers, heart disease, pulmonary disease, hearing loss. And we've made associations between those outcomes and the treatments that they previously received. But we can't go back to the beginning. And by not being able to do that, we have a limited ability to understand what are the mechanisms that cause a lot of these problems. So I think that's a fundamental flaw kind of in our current system, and one that must be addressed. We need more preclinical research that's focused on how our current therapies damage our normal organs, such as the inner ear and the heart, for example. And that's going to lead to our ability to potentially prevent these conditions in future patients. And it might even uncover ways that we could actually prevent further decline of hearing loss, for example, in current patients, and just give people a better quality of life. And so I just wanted to end with two slides here. One is, how do our past successes create new challenges in clinical trial design? So I would say that toxicity cannot be viewed as something that is only an issue during acute exposure. I think the data we've collected now in the 21st century shows us that long-term toxicity is an important issue. And we must, in our clinical trials, we must start to consider long-term toxicity throughout lifespan is an important outcome. We also need to, I think, need to understand more and optimize our ability to collect toxicity data. What that data is and for how long that data needs to be collected, I think, are areas that are open for discussion, but we need to have that on our radar as something that's important in our clinical trial design. And then, to me, I think probably one of the most important aspects is actually trying to change the culture of pediatric oncology, where we don't present our disease as a snippet in time of you're going to go through 6 months to a year of treatment and this is all going to be over and you're going to go on with your life. That's what I was told. And I don't think we knew anything better at the time, but I think we know now. We don't want to take all the hope away from our patients and tell them they're going to have a horrible life filled with health morbidities. But I think we need to set up realistic expectations for where they're headed and to try to give them the tools to succeed no matter what they face. So I think our trials need to take into account that, and I think we need to present new diagnosis in ways that are perhaps different than we have historically done in the past. And then second, how do these successes change our approaches from this point forward? Because that's really, I think, what we're talking about today. My personal opinion, and I hope it's going to be increasingly shared by others, is that we need more research preclinically that looks at off-target effects. And this is for medicines that we've used for 30 and 40 years, in addition to any new agents that come along. So I think understanding off-target organ effects while drugs are in their preclinical development is enormously important, because I think it'll help us design better clinical trials. We need more preclinical research that is focused on how prospective new medicines cause toxicity throughout lifespan. So right now, I think we have a lot of acute toxicity endpoints as something that we focus on for when we move a drug into clinical trials, but we need to develop models, for example, mouse models, which is one of my areas of expertise. You can study lifespan studies in mice over about 2 years. It's a lifespan of a mouse. And so is there a role for those types of studies in the development of new drugs? And then finally, I think very important is comprehensive patient tracking systems. So I think that these need to be established so that we know that true outcome of all of our patients. Some of you may know what the Childhood Cancer Survivor Study is, the CCSS. This was a retrospective study that was one of the cohorts that I referred to earlier, where we assembled patients years after they completed their treatment. But I think we need something like a prospective CCSS. These are expensive, they cost money, but I think it's a priority, because I think it'll allow us to track our patients over time and truly know what's happening to them. And I wanted to end with a quote by Albert Einstein. It says this: "Problems cannot be solved with the same mindset that created them." There was a certain mindset that got us to this point in time where cancer is now 80% survivable. It's created a new problem. I'm not so sure the mindset that got us to this point in time is what's going to be able to help us alleviate some of these untoward late effects, such as hearing loss, that I think impact everybody's life so much. And I just think it's really important to have this meeting today, and I truly appreciate everybody's perspective and what's been said thus far. And I know there's going to be a lot more great comments coming forward. Thank you.